Graptolites are some of the most common and also most useful of fossils from the Ordovician and Silurian periods. But they tend to be overlooked, and that would be a mistake. People don't pay them much attention because they think they're well studied and they're just graptolites. But in fact, these things hold some remarkable mysteries that we're going to delve into a little bit today as I introduce a wonderful group of fossils that deserves a lot more of your attention. Some of the first fossils that I ever found were, well, blades of grass. At least, that's what I thought. I was about 7 or 8 at the time, and it took me until I went to university to finally discover what these things were. You see, my grandmother lived in Llangamach in the middle of Wales, and these are Silurian rocks around there, mudstones as it happens, and they are full of these little sort of black lines, effectively. They look like blades of grass. At first glance, they don't look like much. Even Linnaeus wasn't impressed, which is why he called them graptolites, meaning writing on rocks. Basically scribbles. To him, they weren't even real fossils. And fair enough, I said. But my wife is a specialist on this group, and they are the most wonderful and magnificent animals in the history of the world, except for sponges. And in fact, they do have a lot of extraordinary features, which the more you get into them, the more you discover how much more there is to understand and appreciate about them. Let's start with the basics. We know from their distribution patterns around the world and through different environments, and also from the complete lack of any sort of holdfast, that these things were planktonic. They were floating around in the sea in their billions, trillions. I think someone did a calculation, it was a large number. They really appear in the earliest Ordovician and carry on through to the early Devonian. Although they do have bushier relatives which do have holdfasts of various forms that you find from the Cambrian to the Carboniferous, and even later in some cases. But the ones we'll focus on today are the familiar ones, which are the planktonic graptolites or graptoloids. At first glance, what you see are these little black lines, sometimes with a ragged edge. If you look closer though, that ragged edge is actually surprisingly regular. It is a series of saw teeth, like a hacksaw blade. And when you then appreciate these things are flattened, you start to realise that there's more to it than just an irregular edge. And in fact, we are lucky enough to find ones in different orientations and even three-dimensionally preserved graptolites that can be dissolved out of limestones. And so you have their original form, and then you realise that these saw teeth are just the flattened compressions of a series of little tubes or cups with open holes at the top. A fossil consisting of a series of little tubes like that normally indicates that you've got a colony of animals of some sort. And the problem is that there are lots of different types of colonies, so we need more information to work out what sort of animal it was. And the next thing to notice is that they are preserved always as carbon. Sometimes they're altered to something else, but they are pretty much always preserved as carbon films. So this was an organic skeleton, an organic tube system, rather than something made of a biomineral. There's never any trace of calcite or silica or anything like that. So we have a colony that builds its tubes out of organic material, which narrows it down to rather a lot. Hydroids, most bryozoans, there are many different candidates. So again, that's not enough to pin it down. To get to the bottom of it, you need high-powered microscopes. At that point, what you see is that these tubes or cups are made of a series of little rings pasted on top of each other, or more to the point, half rings, with a sort of zigzag suture running between them. And that is diagnostic. You need electron microscopes to really do them justice, but when you do, you see that there's also bandages pasted over the surface of this array of rings, and the whole structure actually then becomes quite familiar to anyone who likes really obscure invertebrates. Terebranchs are not the most familiar of marine invertebrates, but there are 2 living genera that inhabit tubes, and one of those, Graptopleura, can be found even around the coasts of Britain. Raptopleura itself has a very patchy distribution pattern if you look at their published maps. And it seems to be focused particularly around wherever there's a marine biology research station. What are the odds, eh? Very strange. Anyway, their colonies are little tangles of tubes that encrust onto the surface of a hard substrate, normally a shell or something like that. They can't cope with being exposed to the air, drying out. They also don't seem to be able to cope with being in rock pools even. Maybe salinity changes are too much for them. They're basically very delicate little things. And so you actually have to go diving, normally into deep harbours and places like that, before you'll have any chance of finding one alive. But they are there. The animals themselves are little blobs of jelly with feathery filter-feeding arrays that are all connected to each other within a colony by a little sort of organic thread called a stolon. Their tubes are haphazard, they occasionally branch and then become a bit more of a tangle, but they have no definite shape to them, and they adapt their colonies according to whatever they're growing onto. The other genus is Cephalodiscus, and this tends to be found in much deeper seas and can also become much larger. Both genera have a minor fossil record, with a scattering of rare occurrences going back a long way. The oldest Raptopleura is actually Cambrian in age, and these in some cases even have the individual animals, the zooids, preserved with remarkable detail, so there are some extraordinary fossils. Remarkably, those little animals look almost exactly like they do today, as if the things that we have now have changed very, very little since the Cambrian. They have a fossil record then, but it's very sparse. Raptopleurans and Cephalodiscus just have a scattering of examples throughout geological time. Going back to the Cambrian. And where do pterodactyls fall in animal evolution? Well, they're hemichordates, which means they're surprisingly closely related to us. In fact, the other group of hemichordates is the acorn worms, the enteronusts, and these are, as you might expect, worms, but they also have gill slits, and these and the pterodactyls have a dorsal nerve cord, which is the same structure that we have down our backs, which have been now reinforced with a spine. So there are similarities when you get back to the fundamentals of the body plan with vertebrates. And we're in the same part of the tree. They're all deuterostomes, which is this one division of animal life. So yeah, surprisingly close to us. So we know what these are, we know what they're related to, and we know what the modern relatives look like. But these planktonic graptolites are a whole different kettle of fish when it comes to looking at the detailed morphology, the structure and form of them. And most of this is probably down to adaptations to live in the plankton. So let's have a close look at one and see how it's put together. This one here is Dicelograptus cambriensis from right here in Llandrindod. It's from the earliest part of the late Ordovician, and it consists of a V-shape with two branches, each with a series of cups, thecae, pointing out to the side. All of the interesting stuff in graptolites mostly happens at the proximal end, we call it, the point at which the graptolite first started growing. And if we zoom in on this, you can see that there is, between the two branches, a single conical theca with a spike pointing downwards and a little hair coming out the top, which we call the sicula. This was the original progenitor zooid of the colony. This was swimming around on its own to start with, having sort of metamorphosed in the lava, and it is this that then buds to form the next stage of the colony. So the next little cups that form next to that are the first thecae, they then bud to form the second, and so on. And gradually the colony grows. The form of these thecae is actually quite precisely defined. So in many species there are spines coming off in particular places of particular lengths. Sometimes there are hoods or other sort of structures around the openings of the thecae. The nema, this is a little rod that comes out the top of the sicula, and the vagella, which comes out the bottom end of the sicula. These can have modifications, spines branching structures, even complex veins. And all of these are actually well defined enough and consistent enough that we use them to define species. This is something that is not random, it is hardwired into the construction of these colonies. A lot of these features are thought to be adaptations towards a planktonic lifestyle, which is why we don't really see them in the same way in their relatives that live on the seafloor. And this has even been tested using little wire models in swimming pools or laser Doppler anemometers, whichever takes your fancy or your funding stretches to. And so we know that they have effects. So these spines create little vortices which allow currents to bring food directly into the zooids that live inside the thecae. So there are lots of fine details like this, and it is all, as I say, precisely constrained. All adaptations towards living a life floating around in the surface waters. We actually know the first species that became planktonic at the base of the Ordovician. I mentioned before these bushy relatives that lived on the sea floor. These are called dendroid graptolites, dendroid meaning tree-like, and this one in particular is a thing called Dictyonema. We know these things were benthic because they have a holdfast. You can actually see where they were attached to the seafloor. Okay, that's easy enough. There are many species of Dictyonema. This is another one from the same paper, which, well, Lucy doesn't even know that I'm doing this one, but I couldn't really resist it. It ended up being named after her because she was collecting on that fieldwork in Australia back in 2002, I think it was. And yeah, so you get involved in the research and you contribute something. Eventually, somebody names a species after you. Didn't have to wait long in this case. Anyway, that was Dictyonema. This, however, is Rhabdinopora. Rhabdinopora flabelliforme. And it looks basically identical to Dictyonema. It has the same structure of the branches with the little desepiments joining the branches together, but it doesn't have a holdfast. It just has a sicula, that first zooid, first theca, at the origin of the colony, with a little nema sticking up, and it appears all over the world in large numbers in a wide range of different habitat types very, very rapidly at the start of the Ordovician. From that point on, Graptolites basically went a bit mad. For the next 100 million years they evolved into thousands of species, often quite spectacularly ornamented ones, but with an overall trend of starting at this big bushy thing with lots of branches, gradually reducing the number of branches through time. So through the early Ordovician you went from the sort of ones shaped like that to ones where they flattened outwards, reduced the branches to 32, 16, 8, 4, 2, and eventually you end up with a single branch. In the Ordovician, these were consisted of a single branch with two sets of thecae back to back, joined together. But at the end of the Ordovician, those basically mostly went extinct and they were replaced by a single branch with a single line of thecae. About as simple as you can get. However, there's also a parallel trend in graptolite evolution, in which they keep re-evolving multiple branches. So you have things like Nemograptus, and Certograptus, and Linograptus, many things which are convergently re-evolving a morphology which is vaguely similar in being multi-branched to these ones earlier in the Ordovician. And it turns out that there always seems to be a place for these in graptolite evolution. It's just that every time there is a mass extinction, or an extinction of graptolites, the ones that survive are the smallest and the simplest. When conditions get better, they start re-evolving more complex forms again, and then they get knocked out again, and once more it's only the small and simple forms that survived. It turns out there were actually a lot of extinctions that affected graptolites, or graptolites and some other plankton. So basically, in a mass extinction, it's a really bad idea to be plankton. I don't recommend it. But this means that throughout the Silurian, for example, there were half a dozen extinction events in which graptolites were really badly hit. And so you end up with this progressive trend towards smaller and simpler things, until they eventually died out completely in the early part of the Devonian. But because they were planktonic, and because they evolved so rapidly, and were so badly affected by changes in sea level, temperature, and so on, you have a very rapid sequence of species. Their evolution has been very carefully studied. We have numerous people around the world have documented which species occur where, in what sequence. And so they make perfect species for dating rocks. And it's because of that that we can take a graptolite like this one here, Didymagraptus murchisoni. You find this abundantly at Castlebank, for example, and we know that that is in the Didymagraptus murchisoni biozone of the Darwinian of the Middle Ordovician, and is around 462 to 461 million years old. It's actually a really accurate and precise way of dating almost every sedimentary rock laid down in the sea during the Ordovician and Silurian. As long as there are graptolites there, you can get a very precise date. This is what we call biostratigraphy. So the age in millions of years, of course, it doesn't tell us directly, but we can correlate the bands where these graptolites are found to sections where we have graptolites and volcanic ash beds, and date the volcanic ash beds directly. And at that point, you have a system where all you need to find are the graptolites, and you can say how old it is in millions of years. And that's basically how we date rocks in most cases. So yeah, I'll probably have to do a whole video about that sort of thing and all the subtleties of it at some point. But take-home message here is that graptolites are really, really useful. Always have a graptolithologist on hand. They're really quite handy. It might sound from all this that we actually have a really good handle on how graptolites worked, and there aren't many big questions left about them. But that actually is not true at all. People have studied them intensively because they're so useful, but what they've studied is mainly the biostratigraphy. So we don't know so much about the ecology. There's been some work done on that. It looks like different graptolites lived at different depths within the water column, for example. But there are some really big unanswered questions, which I just want to touch on here because they're so much fun. Question 1 is, why are they preserved in such abundance at all? Rhabdopleurans are not preserved very commonly in the fossil record, and we know what their skeleton is made of, their tubes. They are made of basically forms of collagen. Same sort of stuff as your skin, which doesn't have a very good fossil record, I can assure you. So why are graptolites preserved in their billions? Not only in anoxic black mudstone, where you can imagine decay might have been quite slow, but you also get them in limestones, in sandstones if they haven't been sort of completely destroyed by being washed around. There is something really weird about the composition of these tubes that allowed them to fossilise en masse. And people have touched on this a little bit. There have been a few papers looking into it and suggesting that there is something about their composition originally that gave rise to polymerization processes that stabilize the carbon, lipids being involved possibly, but it's actually an unanswered question, and it's something we don't understand about the preservation of them and what they were originally made of, because their fossil record is so much better than that of rhabdopleurans that we can probably assume that they were made of something a little bit different. And does that relate to their ecology, to their lifestyle? We don't really know. It's a bit of a mystery. But that doesn't even begin to touch on the weirdness of the second one I want to mention. And that question is, how do they even make their colonies in such a well-constrained way? You might not think about it particularly, but this is not a single organism secreting a skeleton that has consistent features, which is genetically controlled by the expression of particular genes in particular bits of the body. This is nothing like our skeletons. It's nothing like a trilobite skeleton. This is a colony. So there was an original animal in the sicula, which then budded. to produce another zooid, which then budded again. All of these are clones of each other. They are genetically identical, but they are separate organisms. So how does, for example, the third clone on each side know to produce a much larger spine, and the next clone goes back to not producing a spine? It's not even as if it's direct genetic expression, because these animals are not secreting it like we do our skeletons. They are externally secreting it. They are using this cephalic shield, a sort of hood-like thing, to latch onto the edge of their theca and secrete another layer. It's more like, what shall we say, the cave swiftlets, which are famous for bird's nest soup, which actually use saliva to create their nests. They're spitting out bits of material that gradually build up into a nest. And yet in this case, rather than building a sort of structure which is controlled by the environment around them and the brain of the bird, who decides where to build the nest and exactly in what form, these are presumably genetically constrained and highly consistent controls on secreting all of the fine detail of these skeletons, these tubes, which we can then use to recognise consistently the species. The more you think about this, the weirder and weirder it gets. Colonies of other animals don't really do that. If you look at hydroids and bryozoans, for example, or even rhabdoplurans, they have basic controls. They have a tube-like structure which is consistent in its overall form, and then each individual within the colony applies that template. So a rhabdopluran has a little tube which eventually buds and branches. But the exact form of that tube, where it goes, the shape of it, is not constrained. So each colony is always slightly different. Bryozoans the same. Each colony is not precisely identical. They have similar branching patterns, they have similar sort of sequences of after three or four tubes, maybe it branches again, but they're not precisely identical at all. There's no mechanism for making them precisely identical. And when you have a structure like a graptolite with complex skeleton with spines, which is precisely identical, we don't know how they do that. Okay, this is beyond my pay grade. I think we're going to need an expert. But how do I get hold of one? Graptolites are rubbish. Oh, hello. Come on in.
Hello.
I found an expert. So, Lucy, your
Yes?
adoring fans would like to know.
Well, both of them.
Maybe all three.
Right, gosh.
How exactly does a particular graptolite zooid within a colony, say the fourth one, know that it has to produce a spine of a particular length, and the fifth one know that it doesn't, given they're all clones?
That's an excellent question. We as Graptolite experts have absolutely no idea.
Oh.
Mm-hmm.
No idea at all?
Well, no, not really. Adam Robanek, who was a Polish Graptolite worker, passed away a few years ago, did think about this in terms of the sicular zooids, so the original zooid of the colony, the one in the sicula, producing some sort of substance that would then diffuse through the rest of the colony. And when it got diluted beyond a certain amount, then the zooids would do something different. Because some Silurian monograptors, they start producing one type of theca, and then they end up producing a different type of theca. So he was trying to explain that, and that may work. But in terms of the first three zooids needing to produce a spine and the fourth one not, maybe it's something like that.
But aren't there some graptolites where you get a big spine on, say, the third or the fourth theca?
Oh, probably, yes.
I'll get some photos of them. There must be. Interesting.
Yes.
So we don't really know.
We certainly don't know at all. It's a big mystery.
It is, and it's just difficult to find a mechanism that could work.
Yeah, it is, isn't it?
Yeah. Okay. I'll leave that one as homework. That's your job.
Can I go now?
Go on
Have
then.
you finished disparaging my favourite fossil group?
Oh, absolutely.
Jolly good.
Yeah. Oh, thank you very much.
You're welcome.
Come back anytime.
Why, thank you. What, any time?
Come back at appropriate times. Yeah. I think that sums it up, really. We really don't know. And that's not even the end of it. There are yet more mysteries surrounding graptolites, like about their reproduction, for example, and how on earth they secreted those veins on the end of the nema in some species. All sorts of peculiarities that we can't get into today, or could, but it would be a very long video. But I'll save for another future instalment when we come back to graptolites, because there's so much more that one can do with them. So I'll leave it there for now. And that was hopefully interesting and has hopefully given you new ways of thinking about a group that far too many people just sort of overlook. They don't spend much time or attention looking at because they're small, they're little sticky things, and sometimes when you start really trying to understand the less dramatic of the fossils, the more fascinating they get. So, thank you once again for making it all the way to the end, and I will look forward to seeing you again with more Graptolites.